So I might just begin in a way that addresses a question that I was just asked, which concerns the relevance of physics and the exact sciences to biology and the life sciences. There are two ways in which I can answer that question. First, with the material that I prepared by way of introduction, and then after that, with an example from my own research. What I will do in the next 3/4 of an hour is really to tell you about my own experience, how I began from your very age and reached the point where I am today. By way of very general introduction, however, let me say the following. So very often one thinks of physics as a study of concepts and ideas, and of biology more about facts, more about a description of nature. But what I would like to emphasize to you, there have been great ideas also in the history of biology, all of which you know. It is also conceptual. So it involves thinking about problems as well as describing nature. And in the end, what you'll see, it requires physics to solve those problems. I heard a lecture given, I think 2 years ago, by a celebrated biologist by the name of Paul Nurse, who discovered the basis of what is called the cell cycle, in which cells divide, and then the daughters of the division divide again. He identified 4 great ideas in the history of biology. And if I were to ask you to think of them, I'm sure every one of you would come up with the same idea. So you might just think for a moment before I begin what those might be, what would come to your mind. Here is what Paul Nurse said. First, he identified the cell, which was discovered by Leeuwenhoek, who made the first microscopes several centuries ago. The cell is the unit of all living things. And Leeuwenhoek, who discovered the cell, was a merchant. And it's believed that this very famous painting by the Dutch master Vermeer, called The Astronomer, is actually a picture of Leeuwenhoek. And if you go to a great museum, and I can't recall if it's in Paris or New York where this particular one is to be found, then you will see the man who discovered the cell. Now, the next great idea was, without a doubt, the discovery of genes, and that will form much of the topic of what I have to tell you. And that was due to a high school teacher named Gregor Mendel. He was an Austrian science teacher, and he asked the question whether characteristics can be passed from one living thing to another. And to answer the question, he planted 2 different varieties of pea plant side by side in his garden, and he discovered that the individual plants would breed true. Each would give rise to offspring that looked like the parent and that did not acquire the characteristics of neighbors. The third of these 4 great ideas you know well, and I don't even need to explain in great detail. It's evolution, and this was actually a bicentennial of Darwin's contribution, so you will have heard about it recently. But the last is the starting point for my presentation to you today. It was the recognition that life is based on chemistry, and chemistry, as you know, flows from physics, and that will be the main content of what I have to tell you. Now, there's something funny about this. The person who really was the father of modern chemistry and life science was Pasteur. And in some way he can be credited with the recognition that every aspect of all living things can ultimately be explained in terms of chemistry. Everything from our capacity to reproduce and give rise to identical creatures to the function of our brains, everything can be explained in terms of chemistry. Now, what is funny about this, Pasteur was at the same time responsible for holding back progress in this field for a generation. He asked the question, is the process of fermentation, which was a prime object of investigation and of interest amongst people in the early history of biological science, is fermentation a property of a living yeast cell, which converts the sugar from grapes into wine? Or is it a property of some component of that cell? He made an extract. He broke open the yeast, and he discovered that the broken cells, the extract from the yeast, was incapable of fermentation. And he concluded that it is a property of a living thing. It is something unique to a viable, to an intact, to a whole organism. His influence was so great, and the respect in which he was held so enormous, no one investigated the question again for 25 years. It was assumed that this was a unique property of a living thing. It was only after the turn of the 20th century, around 1905, that a man named Buchner repeated the experiment and got the opposite result. He made an extract from yeast and discovered that it was capable of fermentation, that it was capable of converting sugar to alcohol. And the reason why Pasteur had failed was because the particular yeast that he used was defective. And when the extract was made, it lost activity. So it was only at the turn of the 20th century that modern biochemistry began, the process of discovering the chemical basis of life could finally start in earnest. And then there were particularly great scientists. They were, in fact, Jewish German scientists, Warburg and Meyerhoff and others, who discovered the enzymes, the proteins within the extract from yeast that actually catalyzed, that performed the conversion of sugar to alcohol. Now, my own career has been concerned, like that of many others, with discovering the chemical basis of life processes. And I told you before, and now I will illustrate for you where it all began for me. I was a student exactly your age, and I had the good fortune to have an introduction to chemistry in high school that was unusual even today. Most chemistry classes, those that my children have taken recently, for example, in the past couple of decades, concentrate on learning a very large body of knowledge about chemistry. Some may find it interesting. Most do not. My kids never wanted to hear anything about chemistry ever again, and I haven't been successful in reversing that damage. But in my case, the chemistry course was, as I say, unusual because it consisted only of a series of simple experiments, very simple experiments. And the first experiment that was performed on the first day is the one that, with the kind help of one of your colleagues here, I can perform for you now. So what the instructor did on that first day was to take 2 measuring cylinders, and he placed crystals of copper sulfate. And some of you will know that copper sulfate is blue, copper ion is blue, and he placed crystals of copper sulfate in the bottom of these measuring cylinders, just as I'm doing now. And then, after that, he poured water carefully into the cylinders. So I'll pour it just carefully down the side. And the same with the other one. And then finally, he mixed one of them. So I'll do that now. And this was essentially the end of the experiment, which you'll see. You may know that copper sulfate is very soluble in water. And when it dissolves, it gives beautiful blue solution. And I'll let the few remaining crystals settle out so you can see. And this is what he showed us. The dissolved copper sulfate on my right, on your left, a beautiful blue solution. And on the left, without mixing, water standing above solid copper sulfate. And then he placed these two cylinders in a cupboard. Asked us to guess, as I will now ask you, how long will it take, given how soluble is the copper sulfate, for the flask on the cylinder on the left to look like the one on the right, if I just let them stand? So what do you think? How long do you think? So that's an answer, never. 1 hour. An hour, okay. A day, okay. So that's helpful. So we have a range of guesses. An hour, a day, or never. We forgot about this because he went on to do other experiments. And on the last day of the class, after several months, he opened the cupboard, and the two cylinders looked almost exactly as they do now. So the answer is not an hour and not never. If you leave them long enough, they will eventually look the same. But what it taught me and the rest of my colleagues in the class, the process of diffusion is extraordinarily slow. Now, we know that gas diffusion is very rapid. Gas diffusion in a room like this is so rapid you can barely measure it. If you introduce something bad smelling like hydrogen sulfide on one side, you'll soon smell it on the other side. But diffusion in liquids is extremely slow. In fact, it would take many, many months for the flask, the cylinder on the left, to look like the one on the right. And this taught me a principle. And indeed, every experiment that was performed in that class based only upon experiments was equally simple and taught a principle. And I think that I benefited greatly in that what success I've had in life as a chemist is attributable to the principles I was taught as a high school student. So the first thing I want to convey to you, it's very important what you're doing now. If you enjoy and you pay close attention and try and perceive what are the principles that you learn, they will serve you throughout life, and as I will now tell you, they may determine the course of your career. One of the members of your group asked, how did you choose what you do? And the answer I gave, it chose me, and this is the very point that I now make. I pursued an interest in diffusion for the rest of my life. Now, after I finished high school and went to college, there I got interested in the measurement of diffusion, especially how one could measure diffusion in liquids. And one of the ways that is done is by the technique of magnetic resonance. Some of you will have heard of MRI, which is used as a non-invasive method for diagnosis in medicine. The underlying principle of magnetic resonance was discovered by Isidore Rabi, a great Jewish physicist who emigrated to the U.S. and did his research at Columbia University in New York. And there's a great story about Rabi, which I have to repeat to you. He used his discovery of magnetic resonance to measure the actual values of the magnetic moments of atomic nuclei. And these had never been measured before. And one day after he had made such measurements, he was sitting on the New York subway going home, looked around and saw all the other passengers and thought to himself, I'm the only person alive who knows the value of that magnetic moment that I just measured. And in a way, that sums up the satisfaction that is to be gained from doing science. Well, magnetic resonance was further developed by Felix Bloch, who is another great Jewish physicist who emigrated also from Europe and was a professor at Stanford, and from whom I learned about magnetic resonance, and Edmund Purcell at Harvard University, who actually taught the electricity and magnetism course that I studied as an undergraduate. Based on their work, it became possible to use magnetic resonance to measure diffusion in liquids, such as we have just discussed, such as I just attempted to illustrate for you. And then for my own graduate study, following the university experience that I mentioned, I then decided to investigate, using magnetic resonance, diffusion in the membranes of cells. So some of you will be aware that the membrane surrounding every cell of your body and every cell in every living thing is made up of two layers of what are called lipid or fatty molecules that have two parts. One that is symbolized by this sphere, which is like copper sulfate, soluble in water. And the other part is a hydrocarbon. It's like the oil of gasoline gushing into the Gulf of Mexico or what have you. It is something that repels water, doesn't mix with water. And because cells are surrounded by such a double layer, and this is only a portion, and it would wrap all the way around the cell, then the cell can be in contact with water on the outside. It can have a water-filled interior, but there is a layer of fat, of what I call lipid, that provides a kind of barrier to the movement of molecules from one side to the other. In that way, defines the existence of the cell, isolates it from its environment. Now, at the time when I began graduate study, in the late 1960s, so about 50 years ago, 45 years ago, it was just at that time appreciated that such a double layer of lipid molecules is the organizing principle for the membranes of all cells. And it occurred to me as a student to ask about the diffusion of the molecules within this double layer. How rapidly do they fuse laterally within one layer of the two? How rapidly, that is, do they change places with their neighbors? And how rapidly can they pass from one side to the other? I chose the term flip-flop to describe that because actually it's a term that comes from physics, which I had recently studied, and I thought that it was an apt term to account for or to describe the process whereby a molecule on one side would change places with one on the opposite side. I used magnetic resonance, and I discovered that lateral diffusion is exceedingly fast compared with flip-flop. And one young person suggested that diffusion in the case that I illustrated might never happen. Flip-flop almost never occurs. The reason is that the salty portion of the molecule that likes to be in contact with water can't traverse the barrier of the lipid and pass to the other side. On the other hand, the process of diffusion within the plane, in, so to speak, 2 dimensions, is comparatively rapid. That is to say, it is at least as fast as molecules will diffuse freely in water, much less rapid than gas, but nevertheless much faster than flip-flop. Okay, so that concluded my graduate experience, and these features of diffusion in membranes are universal, and they are one of the important principles you may learn about cell membranes. They describe the dynamics, not just the structure or the organization, but the actual evolution of membrane properties over time. Now, having studied this problem by one important physical method, magnetic resonance, I decided the logical thing to do to continue my career would be to learn about the other important physical method for investigating life chemistry. So there are really 2 methods from physics that have particular value for understanding the structure and the dynamics of the molecules of life. One is magnetic resonance, and the other is diffraction, especially X-ray diffraction. It can be thought of as a form of spectroscopy. It involves shining light and then measuring what is scattered back. Only in this case, the light is a very small wavelength. It is X-rays. And it permits one to deduce from the pattern of what is scattered back the molecular structure, even the atomic structure of living things. At the time, and this was in the early 1970s, the place to go to learn about X-ray diffraction was Cambridge, England. The first determination of the atomic structure of proteins, so of the active component of living things, the enzymes to which I referred that were discovered by Warburg and Meyerhoff, starting from the work of Buchner. The first structures of proteins were determined in Cambridge, England, by John Kendrew, who, by the way, was also Jewish, and Max Perutz, who was an Austrian Jew who converted to Catholicism to escape persecution in what was to become, of course, Nazi-occupied Austria. These people invented what we call protein X-ray crystallography. Proteins are the most important and the active constituents of living things. So, as I say, I thought I could complete my preparation for a career in chemical science by adding to what I had learned about magnetic resonance, now the subject of crystallography. When I arrived in Cambridge to work with a younger crystallographer named Aaron Klug, who some of you may know of, who was a South African Jew who emigrated to England and then eventually settled in Cambridge, and who won the Nobel Prize for Chemistry in 1981, I had to find a problem to investigate by X-ray crystallography that would serve as a basis for learning the approach. And the research group of Aaron Klug was focusing on structures of viruses and also structures of tRNA that one of you asked about a moment ago. That work was quite far advanced, and it wasn't obvious to me how I could contribute because they were almost done with solving those problems. So I looked for an alternative. And I found what I chose to do in a paper written by Francis Crick. Some of you will know that Crick with Watson, discovered the double helical structure of DNA. Crick was still active in the laboratory, the same laboratory in Cambridge. He was actually a graduate student of Perutz, one of the two people I mentioned invented crystallography. He was in many ways a colleague and a mentor of Aaron Klug. And at that time, at the beginning of the 1970s, he was interested in the problem that I told you I chose eventually, but almost by accident of this experience, for my own life's work. How is the information in DNA expressed in the form that we observe of a complex multicellular organism, such as a human? At that time, in the early 1970s, Crick made a proposal for the organization of DNA in living things. It was published in Nature magazine in 1971, and it's an interesting article to read. Almost no one knows about it because almost everything in it is wrong. And so it was soon forgotten. But someday you might enjoy going back because it's not difficult to read. It attempts to derive this important principle of genetic material and its organization in living things from first principles. And the logic is sound, and the argument is ingenious, but certain of the assumptions which seemed correct at the time proved later to be mistaken, and it led Crick to the wrong answer. I read his paper and thought if such a proposal was made, then it must be tested. And that became the purpose of my own work, which led, as I say, to a very different conclusion from what he had proposed in his own study. Now, I thought I would just introduce this subject very briefly with a background that I believe most of you will know, but just in case, if some of you have studied primarily physics, for example, or mathematics, you may not be aware. So a few slides then by way of introduction to the rest of what I'll tell you. So first, as I think you all know, all genetic information resides in our genes discovered by Mendel, and the chemical composition of genes, the material of which they are made, is DNA. And the way in which the information in DNA is expressed so as to explain the form of our bodies and function is first through copying into a related chemical form, RNA, and finally translation, as it is said, into protein with the use of the transfer RNA molecules that couple to the amino acids, as we spoke of a moment ago. Now, I'm going to show you at the end of this time how the process of copying from DNA into RNA occurs at the atomic level. Today we also know from the work of Ade Yonat at the Weizmann Institute and others how the second step of translation from RNA into protein occurs also at the atomic level. In both cases, the information comes from X-ray crystallography. And I might mention now, and I will emphasize later, the possibility of deriving all of this information, both my own work and that of Ade Yonat and others, derives from a generation of remarkable efforts by photon physicists, and I'll come back to that in a moment. Now, the DNA is the double helix, which I guess most of you are aware of. I'm made of two chains of building blocks, which are these vertical portions. And then it is copied into RNA by an enzyme that I'm going to show you in atomic detail called RNA polymerase. This name actually was invented by my father, who studied the enzyme that copies DNA into DNA for the purpose of heredity. And he referred to such enzymes as polymerases because they are enzymes, thus the designation ASE, that act upon polymers, and in this case, which synthesize polymers. The RNA polymerase copies one strand of the DNA into a double helical form which has both RNA and DNA components. The RNA is in blue and the DNA in black. This is called a hybrid, and I'll show you the actual picture of the hybrid in the course of expression of genetic information in a moment. Finally, for translation into protein, the linear sequence of the building blocks of RNA is translated into a linear sequence of amino acids in proteins, which then folds upon itself in the manner that can be illustrated here, which is the more customary form that you'll see later. And it is the precise shape of these folded forms that is responsible for the specificity to which I referred before when someone asked, how is the right amino acid attached to the correct transfer RNA? Okay, so now let us come to the question that was addressed by Crick and to which I return. We know that the DNA, for example, of a human is about a meter long. There is a full meter length of DNA containing all of the genetic information for construction of a human being in every one of our many trillions of cells. The 1 meter length of DNA must be condensed, must be somehow packaged into a cell on a dimension of only 1 micrometer. And if I were to ask any of you how that might be accomplished, well, the logical answer, if you imagine the very long thin molecule of DNA, is to wrap it around and around like a ball of yarn or sewing thread. Only that creates another problem that relates to the expression of the genetic information. I told you that every cell of your body contains the complete information. Every cell contains all the DNA. How then do the cells come to differ from one another? Blood cells from nerve cells, from muscle cells, and what have you. And the answer is that only a portion of the genetic information is used in each of the different cell types. Now, if the DNA is wrapped like thread around a spool or yarn around a ball, then the problem arises how to gain access to a particular gene in the middle that may be needed in a particular type of cell. So here you see the condensed form of the DNA in cells undergoing division. So this is at mitosis, when there are 2 identical sets of chromosomes that have been made by copying, by the process of DNA replication, and they are moving towards opposite poles of what will become the nuclei of the daughter cells of the division. This is a nucleus of a cell after division. The solution that I found from doing X-ray studies in Cambridge, England, in the early 1970s is illustrated here. So the DNA of every cell is indeed wrapped like sewing thread around a spool, only there is not a single spool but many small ones. Mini spools, if you like. These have been given the name nucleosomes, and they are the fundamental particles of all chromosomes. So the solution is wrapping, but then to avoid the problem that I mentioned before, there is not a single spool, but there are many, so that it is possible to unwrap one without effect upon the others. The way in which these small spools are condensed to produce the form of the chromosomes that I showed you during cell division is illustrated here. They coil up again, and then those coils coil further. There are many levels of coiling to achieve high condensation. Now, the process of packaging in this way itself helps to determine, to regulate the expression of genes, to determine which ones are used in which cells of the body. We now know that it is the selective uncoiling, unraveling of these mini-spools that contributes in an important way to the selectivity of the expression of genetic information in one cell compared with another. But that's not the whole story. After the DNA is made available by unraveling one of these spools, then it must be transcribed by the RNA polymerase to make RNA. And there is also a crucial decision made at that point. Not all DNA that becomes available is read at the same rate, at the same time. The level of the RNA that is made also matters. So what is fundamental to understanding how a single cell gives rise to a complex organism is first to know about the selective unraveling of the DNA, and then after that, the selective transcription by the machinery, the protein machinery for that purpose. And after I left Cambridge, England in 1975, having completed this work, then I devoted the rest of my career up to the present time and into the future to this problem of how is the information in the DNA transcribed, how is it read for the purpose of controlling body form and function, and how is reading the information by RNA polymerase accomplished in a controlled fashion. Now, to solve this problem, I came back to the very thing I told you that I discovered as a graduate student, which in turn I was inspired to study from my high school chemistry, from the experiment that I just illustrated for you here. The requirement for determining the atomic structure of a molecule by X-ray crystallography is precisely what the name implies. It is crystallography. It must be performed on crystals. One must have a three-dimensional organized array of the protein molecule to discover the arrangement of atoms within it by the method introduced by Kendrew and Perutz, and which I learned when I went to Cambridge in the early 1970s. The problem was the machinery for reading genetic information was so large and so complicated that it could not be crystallized for study in the way that Kendrew and Perutz had originally done. And when I realized that one could not, in any straightforward manner, form a large single crystal like those of the copper sulfate that you see here, or salt crystals that you're familiar with, or crystals of sugar that you've seen, when I discovered that one could not form such crystals of the protein machinery for reading genetic information, well, I struggled in the way that I answered to someone's question unsuccessfully, and then finally thought of a way of simplifying the problem, as I told you is one of the ways of coping with such adversity. I decided that I could simplify the problem by reducing the dimensionality from 3 dimensions to 2 dimensions. Only try and make a 2-dimensional ordered array and not hope to grow a full 3-dimensional crystal. And for that, I thought of going back to the discovery I made as a graduate student that molecules in cell membranes diffuse comparatively rapidly in the surface. I spread a layer of those cell membrane molecules, the lipids, with their salty portions and their hydrocarbon portions in a way that's conveniently done at an air-water interface. So the salt part, which wants to be dissolved in water, faces the water, and the hydrocarbon, the oily part, faces air with which it is compatible. And then I adsorbed the protein I wished to study, in this case, the RNA polymerase, to the surface. Now, I knew that if I could adsorb the protein to the surface, it would be constrained in 2 dimensions. And then I hoped that because of the movement, the diffusion of the molecules that I had measured by magnetic resonance in the plane, in the 2 dimensions of this sheet, organization into a crystal might occur. Well, I started trying to do that around 1978. And it wasn't until several years later, after many unsuccessful attempts, through the efforts also of very talented colleagues, we succeeded and got crystals, 2-dimensional, single-layer protein crystals of the RNA polymerase that reads the information from genes for the purpose of their expression. It turned out that once one had got single-layer crystals, that was the breakthrough needed to solve the problem, because single layers would then serve as seeds to support the growth of additional layers that could not otherwise be obtained. And a short while after, we managed to produce now single crystals of the RNA polymerase, which, as you can see here, are about 1/10 of a millimeter across. And that is a suitable size for applying the method of Kenderer and Perutz to find the atomic structure. To do so, we had to go to the very intense source of X-rays available at a synchrotron. This is a picture of the synchrotron at Stanford, and the electrons that circulate around the synchrotron that give rise to the X-rays are actually derived from a 1-mile-long linear accelerator, which you see here reaching into the distance. And then they pass around a ring, which is actually beneath the ground here, and emit X-rays of very great intensity and of a suitable wavelength for our purpose. There are synchrotrons in other parts of the world. There's one in Japan, there's one in Korea, there's a couple in Europe, and there is one that will be assembled actually on the border between Israel and Jordan one day before long, if the project called Sesame finally gets completed, which we all hope will happen before long. But it isn't really necessary for that. An Israeli scientist today who wishes to make use of synchrotron radiation for this type of study, such as Adi Yonat at the Weizmann Institute, will, like people all over Europe, go to Grenoble or elsewhere for the purpose. So it will be nice when there is one nearby, but that is not a necessity for the work. In my case, it was handy that it was at the same university, and I only had to go down the road, but nevertheless. And I might just repeat now what I said before. Our success in determining the atomic structure of RNA polymerase and then of seeing the molecule in action as it reads genetic information, for which the Nobel Prize was given, is really due to the work of a generation of photon physicists who built these machines and created the possibility. And I think, in a way, it was really more they than we who were responsible for the success. And in the answer to one of the other questions, it was who asked about the impact of the Nobel Prize. Well, certainly they should have shared, if not been the recipients of it. Nevertheless, my own colleagues worked long and hard. Over a period of another 10 years after those first crystals were obtained, some 10,000 liters of yeast cells had to be grown over that time, and one graduate student went into the solution of the problem. Well, he's not actually consumed in the solution of the problem. This is a graduate student who was lowered into the fermenter to grow the yeast to repair a valve at the bottom. Nevertheless, the RNA polymerase, the machinery for reading genetic information, whose structure we solved, is actually not 1 but 10 proteins, 10 long chains of amino acids, contains 28,000 carbon, nitrogen, and oxygen atoms. And this is what the structure looks like when you're done. Here's a picture of 28,000 carbon, nitrogen, and oxygen atoms. Now, to interpret such a picture, what one does is to follow the path of the individual proteins through this atomic picture. And here is what the path of the individual protein molecules look like. I told you there are 10 separate proteins, and each is here in a different color. And these ribbons show the path of each chain. And this is a movie which actually lets you see and appreciate both the size and the complexity. A view down a central channel there. Here a view from one side, and then we rotate back to what I refer to as a view from the top. Now the last thing I'll tell you is how we could go on from this point to answer the question, where does the gene enter? And where does the RNA molecule that is made exit for the process of reading the genetic information? And the solution to that problem was found by Avi Ganat, who did his graduate work at the Hebrew University and then came to do postdoctoral study in my group at Stanford. Ganat succeeded in crystallizing not the RNA polymerase, these 10 proteins all by themselves, but the molecule as a complex with the gene DNA in action making RNA. What I'll show you in a moment is a view down the central channel of the structure determined by Ganat, showing the enzyme in action. But I'll just, before showing you that view down the central channel, I'll show you a color code to what comes after. So this is a view down the central channel, but now the colors have been simplified. So this is the entire protein that you just saw, but not in multiple colors, only in white, except for this portion that's in gold, as you'll understand in a moment. And then what this slide consists of is a movie, and it's made of only 2 frames, and each frame is an actual atomic structure. The first, the one I just showed you, but now viewed down the central channel. And the second frame is the structure determined by Ganat, by Avi Ganat, of the molecule with the DNA and the RNA, the molecule in action, transcribing the gene. The DNA is in blue and green, and the RNA is in red, and you see it occupies that central channel. And you all may also have noticed this large protein element, colored gold, swings 30 angstroms over the DNA and RNA, serves as a clamp to hold it in place, so the enzyme doesn't lose its grip from the beginning of the gene to the end. Now what I'll do is rotate this around, and then I'm going to remove some of the protein in white to give you a better view. And here you see what I think is the best illustration of the protein machinery for reading genetic information in action at atomic resolution. We see that the DNA in blue and green, the double helix, enters from the right, and then it unwinds near what is called the active center, where the formation of RNA takes place. There, the RNA in red is assembled, and the building block of RNA just added to the growing RNA chain is seen here adjacent to a magnesium ion important to catalyze the process of gene transcription. That building block of the RNA just added is paired to the building block of the DNA that specifies its identity. The DNA strand in blue is called the template. It directs the synthesis of a corresponding and correct RNA molecule. Finally, and this will be the last thing that I tell you, and it just gives you a sense of what is the work to be done and what is the challenge for the future. Well, the challenge is far greater than what has been accomplished. And I indicated at the outset the purpose of discovering the structure of this machinery at the atomic level is not only to understand how transcription takes place, but how it is regulated. What controls the level of RNA made from different genes in different cells at different times in the life of a living thing. What we discovered, my colleagues at Stanford, now some 20 years ago, the molecules called gene activators and others that are called gene repressors that regulate the level, that turn transcription on or off depending upon the requirements at a particular time and place in the body, exert their effect upon the RNA polymerase and associated proteins that I won't tell you about in detail that read the genetic information through an intermediary assembly of proteins that we call, for that reason, mediator. This is a gigantic multi-protein complex. It is made of at least 24 protein molecules. It is more than 1 million Daltons molecular weight. So if the proteins first solved by Perutz and Kendrew were individual polypeptides, individual proteins, the RNA polymerase I told you is made of 10. Now here we have an assembly of more than 20. And as you will perhaps understand from this simple diagram, the molecule, this assembly that goes between the activator or repressor that turn on or off a gene and the RNA polymerase serves as a kind of signal processing device. It is a molecular computer. At every human gene, there are multiple inputs of regulatory information. There will be multiple activators that define the place and the time and what have you and the level of transcription that is required. And there will also be multiple repressors. These all interact with mediator, which then computes the correct signal to deliver to the RNA polymerase, whose structure you just saw at the atomic level in the act of transcription. The challenge for us in the future is to discover now the atomic structure of this molecular computational device and understand how it interprets regulatory information to deliver the correct signal to the RNA polymerase so that a blood cell will appear as a blood cell and a nerve cell as a nerve. So with that, I'll stop and invite your further questions. So it can be about the material that I presented, or it can be, again, about anything of general interest to you, whatever springs to mind, anything you like. Please.
When was the day you or anybody wake up, says, I want to do an experiment. He does that experiment. And after 20, 10, 5, 2 years, he gets a prize, or somebody else understands it, or he himself understands what he does.
So in the case of the work I just told you about the atomic structure of RNA polymerase, the work really began when the idea came to mind of using a membrane molecule spread in a layer, as I showed you at the air-water interface, to promote crystallization. And it was probably, I think it was while I was still in Cambridge studying the structure of chromosomal material, and with the memory of my graduate work fresh in mind on membranes, that I thought of combining the two in that way. So I really started around 1976. And it was not until 1989, so 13 years later, that we first could obtain single-layer crystals of RNA polymerase. So we tried, I tried, and people who worked with me tried for many years, and we tried by many approaches. So then you ask, well, you know, how could you try so long, so unsuccessfully? And the answer to that is, first of all, that we believed that we would succeed, and every failure only added to our anticipation. You know, we felt determined to get there, and so failure was not discouraging. It was actually a source of motivation to go on. And I'm sure you've had that experience where you fail to do something, and that actually gives you an even stronger urge because of the challenge you now sense. You feel not only that you wanted to do it, but now you feel you want to prove to yourself that you can do it. Also, during the time, we developed the method and the approach, so we could make useful contributions. We could do work of some value even before we succeeded finally in creating those first single crystals. Then after that, it was another 11 years before we obtained finally the atomic structure that I showed you. In the time in between, we could once more obtain interesting information, but only partial information. And once again, our determination to succeed only increased with every failure, because it made it even more of a challenge to succeed. I do remember the day, indeed the hour, even the minute, when we knew the problem was solved. That was in December of 1999. And it was at the point where we could see that the computation that was performed to derive from the pattern of scattered X-rays, the actual arrangement of the atoms, was going to converge on a solution. We did not yet have the answer, but the calculation had begun to narrow down to what we could be sure then was going to be a final solution of the problem. And that was perhaps the greatest thrill that we got in the history of the work. But there were many moments of both failure and determination, and of satisfaction all along the way.
Just one more question. Did you say in the beginning and after you succeed in your project that your work is worth the Nobel Prize?
So there are really two answers to that. The first is that it was obvious after we had succeeded, or even before, that such a thing was possible. But I could give you many, many examples of work that is deserving of a Nobel Prize, and many great scientists who might have received one, but who did not. So there's certainly, I think, an element of chance. That's perhaps the reason why the prize has the particular appeal that it does to the popular imagination. It's kind of like a lottery, like a casino. A lot of great work is done, and one will be the winner. It doesn't really matter, you see, because in our own work, as I have said, and in that of all the others who happened to do great work but not receive a prize, really the reward, the personal satisfaction gained from finally solving the problem after many, many years of trying was so great, nothing else really seemed important after that. It's not maybe a good or a bad thing for such a prize to be given. It's probably good for science, because it focuses the news for one day of the year upon science. And so for one day, people may think less about some of the unpleasant aspects of life or what have you, or all that is less constructive. It's important, I think, for science because then the general public, appreciating its significance, will be more inclined to support science. Less good sometimes for the recipients because then a lot of other things may be asked of them, but that's, if you like, an occupational hazard of doing science.
We'll take just one more question from Marius. Sorry, right? Marius.
Please. So I started, well, let me just tell you, I mean, first of all, with regard to science, before I answer your question directly, I started out studying math and physics. I loved the exact sciences, and actually as an undergraduate student at Harvard College, my major subject was chemical physics. And then my graduate work was actually also in chemical physics with a kind of descendant of Pauling and Bloch and Purcell named McConnell, who I think he is still alive, one of the greatest living physical chemists. And it was really only afterward that I shifted the focus more towards the biological side. But even then, as I've told you, my work really involved a very large component of physical science. So I always had a particular fondness for math and physics. I consider myself a chemist, which I regard as, in some way, the most central. And so I would try to convince anyone who listened the most interesting of all the sciences. But that's beside the point. In real answer to your question, I mean, I'm now going to tell you, I actually began as an undergraduate at Harvard declaring a major in English literature. And I actually liked writing and literature so much that I would have gone on were it not for the high school experiment that I told you about, which led to my studying magnetic resonance on the side, not in any course in college. In fact, I didn't do most of the homework. I shouldn't tell you this. I shouldn't tell the students this. I didn't do most of what was assigned to me in college, in the classes that I took, because I was so interested in magnetic resonance, which was not taught as an undergraduate subject, but which I knew could help me understand the process of diffusion. And so I spent most of my time outside of class actually learning about that. I switched from English literature to chemistry only in my third year as an undergraduate. At the same time, the only other thing I did instead of studying, besides learning about magnetic resonance, was, as it happens, music. I got interested as a child. One day I went to a public school in St. Louis, Missouri, in the center of the country. So I'm not sure everyone in this room has even heard of St. Louis. And I was I think a first grade student in St. Louis at the time of a famous decision of the U.S. Supreme Court when schools were desegregated in America. And I left school one spring from a class that was all white and returned to school that autumn to a class that was almost all black. I was almost the only white student in the class because the black students were now allowed to attend that school, and many of the white parents took their kids out of that school and sent them to private schools. Still a problem in America. I was walking through the school one day when I heard what I thought was a beautiful sound. It was of a 9-year-old black girl being taught violin by a black violin teacher. And I went home and I told my parents I wanted to play violin. I thought it was just the most beautiful thing I'd ever heard. She was terrible. I mean, she could barely play at all. I thought it was wonderful. She played a song called Auld Lang Syne, which is an Irish folk song that you may know. And I wanted to play Auld Lang Syne. So my parents went out and got a violin, and from a friend named Martin Kamen. Some people who are physicists here might have heard of Martin Kamen. Martin Kamen discovered carbon-14. He was a great American physicist, also, of course, Jewish and of European origin. And Martin was a very fine musician as well as a great scientist. And as I say, the discoverer of carbon-14, and he should have received a Nobel Prize for that. But Martin's life was complicated because he was blacklisted during the McCarthy era because of his very liberal views. And this created a lot of problems for him in getting funding for science and what have you. So Martin found the violin for me, and I started to play violin. And by the time I was in college, I used to do that 5-6 hours a day. So there's the answer to your question. Am I good at it? That's a matter. That would be a matter of a question, a matter of taste or opinion.
Okay, we would like to thank Professor Roger Kornberg for his beautiful lecture, taking the time and answering the question. I believe we could continue on and on, but we are going to drag in some other things. But I really hope that there will be some other time, we'll come again, and we'll be able to catch on it and continue, and will be some more time to questions, answers, dialogue. So
It's a pleasure.
Thank you so much.